All right, right now we're discussing section 9 of chapter 12 on nervous tissue for anatomy and physiology on the characteristics of action potentials. The objectives for which you should have understanding following this section are to compare the graded potentials and action potentials, describe the two primary factors that influence the velocity of action potential propagation, identify the criteria used to distinguish the groups of nerve fibers, and describe how action potentials vary in frequency. So with graded potentials versus action potentials, the graded potentials occur in the neuron's receptive region due to the ion flow through chemically gated channels. They can be positive or they can be negative changes in charge. They're graded, they have a larger potential change to a stronger stimulus, and they're local. They travel only a short distance. By contrast or comparison, action potentials occur on the neuron's conductive region on the axon due to the ion flow through the voltage gated channels. They involve depolarization with sodium in and repolarization, potassium out, and they're all or none since once that threshold is reached, happens or it doesn't. The propagation goes down that entire axon to the synaptic knob. As far as conduction speed, is dependent upon the axon diameter and myelination. So there's more myelination or a wider diameter, greater speed. Thicker fibers conduct faster than thin ones. Thick axons offer less resistance due to current flow down the axon. And myelinated fibers conduct much faster than unmyelinated ones, with current flow under the myelin between the nodes being very fast. As far as nerve fiber groups, the nerve fiber is an axon and its myelin sheath. Right? Group A, the conduction velocity is as fast as 150 millimeters per second, and large diameter myelinated fibers are the most somatic sensory neurons here. They're all somatic motor neurons as well. Group B conducts at about 15 meters a second, it's about a tenth the speed. And Group C at 1 meter per second, even slower, due to small diameter and or unmyelinated. Some visceral neurons are in this category and some somatic sensory neurons from the skin. As far as the frequency of action potentials, impulse frequency or action potentials per second varies. Although impulse amplitude, the change in the voltage is constant, the firing frequency varies with stimulus strength. Bright lights cause faster firing frequency on the optic nerve than dim lights, as an example. When motor nerves fire at a faster frequency, it causes the muscle to generate more tension to a point, right? For neurons that can use different neurotransmitters, the firing frequency can influence the type of the transmitter released. So what did you learn? You should explain how action potentials differ from graded potentials. You should know what the general characteristics of group A nerve fibers are and what functions they normally serve, although true B and C as well, but primarily A. Explaining how the frequency of action potentials differs from the velocity of an action potential propagation. So section 1210. Neurotransmitters and neuromodulation. After going through this section, you should be able to identify the 4 classes of neurotransmitters based upon chemical structure, describe how neurotransmitters are classified based upon function, describe how acetylcholine functions as a neurotransmitter, discuss the different mechanisms for removing neurotransmitter from the synaptic cleft, define neuromodulation, including its function in facilitation and inhibition, describe how nitric oxide and endocannabinoids function as neuromodulators. So in classification of our neurotransmitters, they're synthesized by neurons, and they're stored within vesicles in the synaptic knobs, the synaptic vesicles, right? They're released from the vesicles when action potential triggers calcium entry in the synaptic knob, just like we have muscle, same way with the neurons in general. They bind to a receptor in a target cell. They trigger a physiologic response in the target cell, and approximately 100 different ones have been named and classified into groups. Clearly, you're not going to have to know all of those. For the 4 main chemical classes of neurotransmitters, which you do need to know, acetylcholine is a primary one, or a primary player, at least I should say. Its structure differs substantially from other transmitters. Biogenic amines or monoamines, they're amino acids, the amine, right? They're slightly modified to synthesize a transmitter. Catecholamines are in this category, like dopamine, they're made from tyrosine. Indolamines, like serotonin, which was misspelled there, are made from histidine or tryptophan. Amino acids, which include the commonest transmitters glutamate, glycine, and GABA. The neuropeptides, which are chains of amino acids that are 2 to 40 amino acids long, include endorphins and substance P. Now, neurotransmitters are also classified by function, and their classification is by effect on the membrane potential. Excitatory transmitters cause the excitatory postsynaptic potential, whereas inhibitory transmitters cause the inhibitory postsynaptic potential. but some transmitters can excite some targets and inhibit others. And it depends on the specific receptor that's present on the target cell at the time. So it depends. Classification by action. We have our direct and our indirect. Direct transmitters bind to the receptors that are chemically gated channels with an immediate postsynaptic potential. And we have indirect transmitters that bind to receptors that involve G proteins and second messengers. The more diverse effects are possible with the indirect. So with our classification of our neurotransmitters then, so we have classification based on chemical structure, the acetylcholine, biogenic amines, amino acids, and neuropeptides. Then we have, based on function, our effects, which are either excitatory or inhibitory, and our actions, which are either direct or indirect. Direct being with open ion channels, and indirect, indirectly causing a response to the activation of G proteins. As far as features of neurotransmitters, acetylcholine is our best characterized neurotransmitter. It's used in the peripheral nervous system to stimulate skeletal muscle. It's also used in the central nervous system to increase arousal in time of stress. Otherwise synthesized from acetate and choline, acetylcholine, right? It's stored in the synaptic vesicles. Its action potential triggers its release into the cleft, and some acetylcholine attaches briefly to the postsynaptic receptor, the neuron that's being communicated to, and the ACH, or the acetylcholine, is then cleared from the cleft by being broken down to acetate and choline by acetylcholinesterase, right, an enzyme that breaks down acetylcholine. Acetate and choline are then taken up by the presynaptic cell for recycling. The effect on the target cell depends upon the receptor present. Nicotinic versus muscarinic receptors are examples there. We'll get some detail more on at a later time. The removal of the neurotransmitter from the synaptic cleft can occur in varied ways. So getting rid of that neurotransmitter in that synaptic cleft can occur either by enzymes degrading the neurotransmitter, or presynaptic transporters might import neurotransmitter, get a reuptake, and then some neurotransmitter diffuses away from the synapse and it's reabsorbed by glia, by the glial cells. Some drugs have their effect by influencing transmitter removal. That's how they get their effect. Selective serotonin reuptake inhibitors treat depression in this way, and galantamine hydrobromide is an acetylcholinesterase inhibitor that's used to treat Alzheimer's as well. So with our acetylcholine release and removal of the synaptic cleft and its action, so we got our nerve signal, and we have our acetylcholine in the synaptic vesicles here. We have synthesis and release. The ACH is release inhibited by botulinum toxin. Remember, calcium had to propagate this initially, or not propagate it, initialize it for acetylcholines released from our synaptic vesicles here. But removal from the synaptic cleft for the get rid of that acetylcholine. Acetylcholine is broken down by acetylcholinesterase, and then we get choline and acetate. And acetylcholinesterase is blocked by nerve gas, and some insecticides can play a role with this, affecting it. And then we have the direct response through our channel here with sodium playing a role here at the nicotinic receptor. And we have our muscarinic receptor here, which is indirect. Okay, so the nicotinic being direct here and indirect with the muscarinic receptor. And that's in our second messenger here with open channel there. This, of course, causes our excitatory postsynaptic potentials here with sodium and the nicotinic receptor. And over here we have activated G proteins playing a role here, causing EPSP or IPSP, the excitatory or inhibitory postsynaptic potentials. These muscarinic receptors are blocked by atropine, and the nicotinic receptors are stimulated by nicotine and inhibited by curare. Now, neuromodulators are chemicals that alter responses of local neurons. As far as facilitation or inhibition, they're facilitated with modulation, then it causes greater response in the postsynaptic neuron, and they can increase the amount of neurotransmitter in the cleft or the number of postsynaptic receptors. They're in inhibition. The modulation causes a weaker response. They may decrease the amount of neurotransmitter in the cleft or number of postsynaptic receptors. You're not going to have to know nitric oxide or endocannabinoids at this time, so not going to worry about it in detail at this moment. So I want to make sure that we don't note that they're there and they play a role, but right now we're not going to require you to know their details. So what did you learn? Describe how neurotransmitters are classified based upon structure and function, and note how it's possible for acetylcholine to generate either an EPSP or an IPSP, and how does nitric oxide act as a neural modulator. And we have our final section with neural integration and neuronal pools of the CNS, identifying the 4 different types of neuronal pools and explain how they function. So our neuronal pools are neuronal circuits. They're groups of neurons that are arranged in specific patterns. There are 4 types: converging, diverging, reverberating, and parallel after discharge. The pool can be localized or it can be distributed in several regions of the CNS. So in this case, we have all of our input coming from our neurons here, and be a postsynaptic neuron here getting output here traveling down its axon. Get a converging circuit with multiple neurons to one neuron here. Input converges at a single postsynaptic neuron from several other neurons, presynaptic neurons. So you get multiple sensor inputs at the synapse on the neurons in the salivary nucleus as an example. Sights, sounds, and smells of cooking lead to one output, salivation. Okay, getting one response from all the different inputs. A diverging circuit, we have one input and to multiple outputs. So we have one presynaptic neuron to multiple postsynaptic neurons here. So we get spreading information from one presynaptic to several postsynaptic, like the neurons in the brain that control the waking send commands to several different muscles for proper balance, posture, and motion. Okay. Reverberating circuit. Here we have feedback that's produced, it's repeated and cyclical activity. So you have input and out to one we have neuron coming in over here, the presynaptic, the postsynaptic here, input this way, and then it could be a cyclical motion here coming from these presynaptic to postsynaptic neurons and cycling back around. Once it started, it stays active until there's an inhibitory stimulus or synaptic fatigue. That's important to know. There are circuits that keep us breathing during regular sleep, as an example. Then the Parallel-After-Discharge Circuit, input is transmitted simultaneously along several paths to a postsynaptic cell. So it's kind of along several pathways coming down till it meets this postsynaptic cell here. And since paths can vary in number of synapses, the signal arrives at postsynaptic cell at various times. So it doesn't come at the same time. And it's believed to be involved in higher order thinking, where you might see a Parallel-After-Discharge Circuit. You should certainly know examples of each, and generally speaking, the general design or how each one generally works. So what did you learn? How are neurons arranged in a converging circuit? And what are the differences between a reverberating circuit and a parallel after-discharge circuit? That was important information to know. And that's the end of our chapter 12. It's not exactly light content. That's why we're going to have one test on one chapter versus multiple this time around.